Oh wait, let me go and do that story. Okay. We'll start.
Okay. What's up, guys? Welcome to the Topics 5.7 Bio 285 review.
I'm Angela.
I'm Keller.
And this is the final exam review. Yay! Everybody clap, yay! Last one. Okay, got three whole lot of topics. Cell signaling pathways. We did a little poll. Gonna breeze through that. We're still gonna do the questions, but we're gonna try and focus more on exam/quiz questions for cytoskeleton and cell cycle. Not that we're not gonna focus on 5 at all, but like you guys did pretty well with that on the exam. So we're gonna briefly do that, but we're gonna try and focus more on 6 and 7.
Yes.
Okay, guys, this is what life is gonna look like after you're done with this final. Okay, here's my story time from when I took this class and I took the final. Whatever crazy things people have told you about this final, it's true. Okay, I'm telling you that right now. Like, it's tough love. Like, I care about you, want you to be prepared. Like, I went in there thinking that I knew everything and that I was the best, and I was not the best. I was not the best at all. And you're gonna reach a point where your morale is so low, and then you have to get up and move to another room because you have to keep taking the exam. And you're gonna panic because you think you know everything, and maybe you don't. And that's okay. And you're not going to do as bad as you think. You're going to walk out of there because it's such a long exam. Your brain is so fried. You're going to be like, I did horrible. I promise you, you did not do horrible. Okay. So know that. Okay. You have the insider information right now. Everyone's going to be like, I'm going to get 100 on this. Like, it's going to be the easiest thing ever. Even if you do the best that you can with studying, it might be hard because number one, you haven't done multiple choice in this class before. You will have multiple choice on the exam. But advantages, you have the 8x11 sheet, both sides. Okay, so you can, as long as it's handwritten, hand drawn everything, you can label the proteins. You just can't say like definitions. So this protein does this, and like you can't do that, but you can have diagrams, which is like a lot of help from going from we don't have cheat sheets at all to being able to have that. So that's good. But you're gonna walk out of there, and this is what life is gonna look like, guys. I promise. When I took it and I went outside and I was visibly like very just like burnt out. And this is before I was like gonna TA for her or any of that. Like I kind of had a relationship with her, but not really. And she came up to me and she was like, so how'd it go? And I was like, oh, not good. And she was like, it's okay. And then we talked for like 40 minutes, and that's how I like got to know Laura. And it was right after I felt like I failed the final. So don't panic. You've done your best, like past a point, what can you do? You did your review, yeah. Multiple choice, okay, I'm not a good test taker when it comes to multiple choice, but I don't remember specifically the multiple choice being that bad. The problem is when you finish the multiple choice and you've used up all that brain energy for like overthinking options, is that now you have to go into the open response, right? So that's like, we're talking about strategy. If I could go back, I would probably do the open response first. For me personally, because I have very low test endurance, I would have done open response first and then gone over to the multiple choice. So it's kind of preference. I'm just saying that for me, if you're better at multiple choice, that's not really a problem. Whatever. But it's tough to do all the multiple choice because I think there's 40, 50?
I think there are 54 multiple choice
50.
questions.
Okay, yeah.
Yeah.
So then after that, then you got to go and do all of the open, right? So keep that in mind for like, how are you going to tackle this? Are you going to do open first? You're going to focus on that? Or are you going to like maybe waste all your brain energy on multiple choice and then be tired? So just keep that in mind.
I would say the multiple choice are like, for the most part, pretty straightforward. There's definitely a few that are meant to be tricky and trip you up. But I think one thing that helped me a lot when I was in the exam is I just kind of left those and then went to the free response. And sometimes the free response questions basically give you the multiple choice answers in the questions. So you can always look at later questions to try and figure earlier stuff out too. Yeah.
Like, never go with the open responses.
I don't even remember. I took this last year.
You just black out.
Uh,
You just black out, really.
Yeah, it's like black hole.
The graphs,
Um,
the graphs,
There are graphs on, there were graphs on it.
the graphs.
She loves
killed me.
graphs.
She loves graphs.
I wouldn't say they're as bad as the midterm free responses, from what I remember. They're definitely shorter, and she includes a couple matching ones too that were more like the ones on the second exam that we took, the midterm. So you don't have to be writing the entire thing, you can just kind of like fill in stuff. They're definitely not as long, I think.
Yeah, I think she purposely, except for the graph one, she kind of makes them a little bit easier than normal, like the open response, because she knows you spend so much time doing multiple choice. The only tricky one that I like deliberately remember from this was literally like 2 years ago is that graph one, which we're gonna help you out with when we get to that unit.
Yes.
So don't worry. Okay, any other questions about like exam format, anything before we get into it? Okay, let's go. All right.
You want this or no?
Yeah,
Okay.
yeah, yeah. You know. All right, topic 5. So we're gonna go low key, kind of fast through this, but there are some concept questions that I'm gonna repeat, and you're gonna be angry at me because you're like, we've already done this before, but they're really important, and I wouldn't be surprised if she put this on your exam. Okay.
And also we have prizes for answering the questions. So that's fun.
You want to say the lineup? You want to say the lineup?
Yeah. We've got some ginger snap cookies, Kinder Bueno stuff, truffles, and peanut butter cups, dark chocolate and milk chocolate.
That's a pretty good lineup,
So
guys.
there you go.
Okay, so super quick, cell signaling basics. We're gonna rush through this. I need all of your attention. Like we're speed running this. Okay. So signals can be extracellular, which is outside or inside the cell. Outside. Or it can come from within the cell, and there's the 2 types of signals, but you guys already know about that. Let's just do this super quick. I hope you like the meme I put. I found this and I thought it was really funny. Okay, if a signal can pass through the phospholipid bilayer, this fun guy right here, it is blank and blank. Who wants to answer? You guys, the whole time. Okay, yeah. Small and hydrophilic. Okay, so which means if a signal can't pass through the phospholipid bilayer, it is what and what?
No,
Oh, wait, you're actually wrong. She was actually wrong. I'm not gonna call her anymore. Okay, so approaching this question. Phospholipid bilayer. What is that yellow part in the diagram? What is that? Lipids. Lipids, right? Lipids is fat. Lipids equals hates water. That's how I remember it. Okay. So signal can pass through phospholipid bilayer. It is. I saw another hand over here. Did you want to answer? Either. For the first one?
Small. One of them is small.
One of them is definitely small. Okay, so if signal can pass through, it is smaller and hydrophobic. Why is it hydrophobic? Because what's that middle part there? What's the yellow stuff? They're lipids, right? Lipids don't like water. So if it can't pass through, it must be larger and hydrophilic. So it can't get past that middle area there, and it is too big to kind of like sneak its way past. Okay, 2 outcomes of cell signaling. I messed this up in the last presentation. It's fixed now. 2 main outcomes. One is altering protein function. What is a function? Like the job, right? So we alter the job of a protein. That is the quick way to do it. The slower way is altering protein synthesis. So altering the way that the protein is, like what proteins we make, blah, blah, blah. So these can both happen, and they're not mutually exclusive. If she ever asks you about that, maybe it'll be a multiple choice question. I don't know. She likes those kind of questions. But can somebody tell me why protein synthesis might be a slower way to do it? Why is it not faster?
Factors, and you also deploy nutrients.
Exactly. So transcription, translation, all stuff that's involved with making proteins, those are processes that take time, right? So if we alter the function of a protein, instead of going from scratch and trying to make more of something or another, that's a way quicker way to do it. But again, these 2 things can happen at the same time. It's not one or the other. This is just the fast way to do it, is altering protein function.
Okay, next we have signal amplification and crosstalk. Crosstalk is when pathways kind of talk to each other. So one protein will give a signal to another protein, and messages can kind of pass between pathways. And signal amplification, which I think some people got a little tripped up on this when I was doing like quizzes and stuff, is when you have one protein targeting multiple downstream proteins in the same pathway. So say you have one protein sending a signal to like 7 different proteins, and then those 7 proteins each target another 49 downstream proteins. It's kind of like exponential growth throughout the pathway. It's not just the signal moving down from one protein to another. Yeah, and the purpose of crosstalk is to coordinate responses in the cell between different pathways so that things occur when they need to at the same time. Yeah. Cool. Okay, so it's like our first official question. What are the pros and cons of having a multi-step pathway compared to a pathway with few steps?
Let's start with just pros. Let's just do pros for right now.
Just pros. I don't know. In the back, yeah,
More crosstalk, more opportunities for crosstalk. Yep.
nice.
That was pretty good. What else? What are other pros?
Oh, yeah, sure. Yeah. Yes, perfect. More amplification. Yeah. Now some cons. Yeah. Oh, if you want to give another pro, that's fine too. Yeah, it's slower signals, right? So each step is kind of slowed down throughout the process. What? Sure.
What did you want to say for a con? Did you have? Yeah. More chance for mutation. Yeah, like so stuff can go wrong, right? Yeah, that's a good one. I didn't put those one, but that's definitely one. Like stuff can go wrong always when you have more things and more variables to keep track of. Does anyone know? Oh yeah. Do you have another one? Yeah. Yeah, it takes so much more effort. So like that's another one I didn't have on there, but that's definitely an answer. Takes way more energy to do.
Sure.
And nobody wants the choice.
Here we go.
Does anyone know what diffusion rate limiting kinetics are?
Yes.
Yes, I actually just put it up there, so I'm I guess not gonna answer. But it's basically, if she ever uses that term, it just means it's the likelihood of 2 specific molecules bumping into each other when they're just floating around. So when signal amplification happens, and like Keller said, it's not just one message being passed down to one thing. It's like gossip. It's like, I'm gonna go tell somebody something, and then they're gonna go tell a bunch of people, and then they're gonna go tell a bunch of people, and now that message has spread. So diffusion rate limiting kinetics is like the likelihood of 2 people that are going to talk to each other. It just means that like we can't control how fast a message goes past some point, because that likelihood of 2 specific molecules bumping into each other randomly when they're just floating around, we can't really control. So like, what's a way that we could like increase signal amplification? Does anyone have an idea? Like, what's one thing that we could do in a pathway to make sure that message gets spread out farther? Anyone have an idea? Yeah. Increase the concentration. More people talking to other more people makes messages spread. What else? What else? This is like the asterisk star one. Yeah, so that's kind of like the similar thing, right? So like increased concentration. Yes, but there's something I'm looking for. Self-small. I mean, yeah, like you'll increase the rate limiting kinetics, right? But what I was looking for was stuff, right? So this is the asterisk. This is what we're talking about in the last exam review. This is what I talked about in the topic 5. This is really important. And any question, when she says, when it comes to signal amplification, she's like, okay, I changed this one thing. What's going to happen? It's whatever the answer is, plus, but crosstalk is always a possibility, meaning any other pathway can come in at any time and turn something on, or one pathway can talk to another and just turn something off, right? So yes, like if we increase the concentration, we're going to spread the message out farther. But also, what if I just like skipped a bunch of steps and I had another pathway just turn on something at the end? Is the message still going to get across? Yeah, I'll still do it, right? So again, if she asks me a question about this, Always, always, always say what the answer is, like, if you're just thinking about it normally, but always go, there's always a possibility of crosstalk. Got it? Cool? Okay, any questions on that? Okay. All right, we're gonna go through the pathways pretty quick because you're gonna have a diagram. But I just want you to get a sense of the flow in your head. So when you're approaching these questions, if she asks you if there's a loss of function or gain of function in something, I want you to think of it just as stepwise as you can once you get those steps down. If one thing is wrong, you can kind of go through those steps and be like, okay, well, if that step can't happen, then that step can't happen, right? That's how you approach these questions, but you guys did a really good job on exam 5, all because of me. It's all because of me, obviously. Okay, so RTK pathway. Signal binds to receptor domain, portion outside cell, which is called what? What is that term outside cell? Extracellular, right? Which causes a conformational change allowing the two RTKs to come together. What is that term called when two things come together to form one unit? Dimerization, right? Two come together to form a functional unit. Tyrosine kinases are then able to cross-phosphorylate or autophosphorylate. Autophosphorylation means you phosphorylate yourself. Cross-phosphorylation, they're just phosphorylating each other. And then basically those phosphorylated tyrosine kinases serve as the binding sites for downstream proteins. Don't have to know much about that. It's called the signal transduction cascade pathway. So that's just like people talking to more people, talking to more people. You don't really need to know about that. Okay, so RAS signaling. Okay, so dark blue guy. See the dark blue guy up there? Adaptor protein. That is GRB2. We're going to go over a question. She talks about GRB2 in, I think it was the quiz. We're going to go over one of those questions. But basically, GRB2 has one side with an SH2 domain. We're going to talk about the domains in a second. And it has another side with an SH3 domain. Actually, it has two SH3 domains. So it actually has both. So what adaptor protein means is that we're just connecting the RTK on the left to the rest. That's all adaptor kind of means in this case. So GRB2 binds to one of the phosphorylated tyrosine kinases on the RTK through the SH2. And then the other side binds the RAS-GAP over there, the SOS, which is that other one's connected to, which activates RAS. We're going to get into specifics in a question in a second. And then because of RAS-GAP, they'll change RAS from GDP to GTP through the shape changes, and GTP means on or off. On. GTP on, GTP is off. Okay? Okay, and then all you have to know about this is that the active RAS activates MAPK3, which does MAPK2, which does MAPK1, 321, pretty easy to remember. MAP kinase cascade, and then basically it just lets MAPK phosphorylate downstream targets. But okay, just know that 321, that's all I will remember.
Good. Cool.
Okay, questions, new question, yay. So this is from Topic 5 quiz. So GRB2 contains both an SH2 domain and an SH3 domain. Describe what SH2 and SH3 domains are and what they do in general, and then discuss what they were binding to and doing in this pathway that GRB2 is involved in. So let's go over what these two are first. Who can tell me what SH2 domains bind to? Fill in those blanks for me. Close phosphorylation. Yes, that is the
Nice.
phrase, right? So SH2 domains are what domains? Phosphor, what's that phrase? Tyrosine kinases, right? Not just tyrosine kinases, phosphorylated. That's important, right? If you can't phosphorylate it, can't open it up, things to bind. That's important. Meaning that they bind to Blake on other proteins. Yeah, that's pretty straightforward. Okay. SH3, you can tell me what SH3 is a domain that binds a Blake on another protein. Who knows what that blank is? You guys, I know you guys are, yeah. PXXP motif, right? Blake are regions of proteins, that's the PXXP motif, are regions of proteins that have several codons and often form helical structure. So SH3 domain binds to PXXP motif on another protein. Yeah? Yeah, sauce is an example of one.
Yeah.
Yeah, so that's right there. So can we do the next one? So what does GRB2 have to do with this? GRB2 is the adaptor protein that activates the RTK with RAS, right? And it has two domains. It has which two? SH2 and SH3. So when RTK is active and autophosphorylated, what does autophosphorylation mean? It just phosphorylates itself, right? GRAP2 can bind to the phosphorylated tyrosines on the RTK, right? Actually, can you go back to that photo of it? Yeah. So dark blue on the left side, it binds onto the phosphorylated tyrosine kinases, right? So that needs to happen first, and then you can go back to the regular slide. Thank you.
Too far.
Yeah. And then the other side that has the SH3 can bind to the SOS. So SOS, the RAS gap, we're gonna get into gaps, all that stuff, binds GRAP2 and activates RAS. So SOS, so the light blue one on the right, it has the PXX motif to grab to get to GRAP2's SH3 domain. Okay, so left side, GRAP2 has SH2 domain, phosphorylated tyrosine kinases. Remember that phrase. Right side, it has the SH3 domains, which bind to what? What do SH3 domains bind to? The SH2 motifs. Who has that? SOS. Make sense?
I'm gonna skip this one because we just did it.
Yeah.
Basically. GDP off, GDP
Yeah.
on.
GDP on, GDP off.
Cool.
Nice. Okay, cell signaling. So, result of this pathway, does the cell survive or die? Survive, right? So make sure you don't get that mixed up. So the end of the pathway is not that the cell dies, the cell survives. So it's constantly preventing apoptosis, which is what?
Program cell.
Program cell death. So a signal, again, we're gonna go this really quick. Signal binds RTK, receptors dimerize, which means what? Two things become. That's after. So the two things become one. PI3 kinase, PI3 is that long blue guy right there. Binds to the phosphorylated RTK through the SH2 domains, because SH2 domains you bind to phosphorylated tyrosine kinases. There's that phrase again. And now that PI3 that's activated, and now it can go and activate, it can phosphorylate PI. So the intensol, whatever that is, that's just PI. PI becomes PIP3, which we just call PIP, PIP3, they're kind of interchangeable.
Yes. Domains.
Oh yeah, okay. PI3. So protein, it has an SH2 domain that will only bind tyrosine, only binds RTK once it's been phosphorylated, because as we know, SH2 binds to phosphorylated tyrosine kinases.
Yeah.
Cool. Okay. So now we have ATK. ATK is central downstream regulator. AKT, sorry, is green guy. So when it binds to the PIP through its pH domain, I don't think it's important that you know it's a pH domain. But what happens when AKT is binded to PIP is that it can be brought to the inner surface of the cell membrane. That's what it does. So it gets bind, the binding brings it there. Okay. Same thing happens with PK1. Also has this pH domain. Also goes to the same area by binding to PIP. Okay. I redid this slide because I think last time it's a little bit confusing, so I hope this makes more sense. But if we phosphorylate AKT once, is that activated? No. No. That's the asterisk, right? You need two different phosphorylations of AKT for it to be fully active. Got it? So what's the first one? PK1 is to phosphorylate AKT. That's the first one. And then the second one, what's the second one? PK2. Pretty easy to remember. What do kinases do? Phosphorylate things. Who's phosphorylating AKT? PK1, PK2. I feel like that's pretty easy to remember. And now AKT is fully active. So if AKT is not fully active, is that signal going to get anywhere? No. So if PK1 phosphorylates AKT, but PK2 doesn't, is it active? No. That's important. Okay, she'll definitely throw this in there, like, what if one of them has lost function? And then you're gonna be like, it doesn't matter if there's another one phosphorylating it because it needs two activations. Okay, yeah.
Does PK1 plus 4 PK2 to the area?
It opens up the site for PK2 to phosphorylate it. Yeah, I don't think it necessarily recruits PK2, but as PK2 is like diffusing around, right, it can't phosphorylate AKT until PK1 opens up the site for PK2 to phosphorylate it.
AKT is released to cytosol.
Yeah, because the PIP domain it's like a thing that kind of slides in the cell membrane, right? So it's stuck to that, attached to the cell membrane until it's fully phosphorylated, then it can release. Yeah, PK1 and PK2 both have to phosphorylate it for it to be able to, from PIP. Yeah. Sorry, what?
Oh, I think you mean like once it's phosphorylated by PK1, does it release? Yeah, okay, so that first phosphorylation is what causes just AKT to be released from PIP, which lets PK2 come in.
Yeah.
Okay, does that make sense? Okay, any other questions? Yeah, we're gonna go in fast through topic 5 because I want to get to topic 6 then. Okay, okay, I want you to turn something for like 30 seconds and just explain this process super quickly before we go on to bad and all that stuff. Go, 30 seconds.
Bad.
Basically just
Speed run.
Speed run.
Literally speed run. Yeah.
Okay.
That'll be pretty quick. And then
I can pick up JackStat since you've been doing this forever.
Okay. Yeah, I hate Jackson.
Okay. I'm
I
fine
hate
with that.
Jackson. Yeah, and then you want to do you want to do these things as well? Or
Sure. Hang on.
okay, cool.
I didn't look through all of it. Just check this out.
Well. Look who it is. Look who it is. Hey, you got a haircut? Why do you get a mustache? Why do you shave it all off? Starting your demon art? Okay, stop. Okay, who wants to quickly like recap for me in like literally 30 seconds? Who wants to do that? Who wants to be brave? Who wants candy? Oh yeah, we have candy, guys. Since you just decided to join us.
Signal binds are the RTPase and then Garmin
Oh, my bad.
That's the binding site for the PI3 kinase with the SH2 domain, and then that allows PI3 to then phosphorylate PI to SNAP-E. That's the binding site for then AKT and then PK1, then PK1 can then phosphorylate and cause AKT to let go, and then PK2, and then it's fully phosphorylated AKT, then that targets the bad.
Yep. Yeah. Whoa, we're not there yet. Okay, that's a whole other thing. We're not. But yeah, that was perfect. Great. Okay. Any last questions before we go on to the second part of this process? Okay.
Thank you.
Okay.
Nobody wants the truffles, bro. Nobody wants the truffles. You want one?
Here, you already got one.
You have a question?
You didn't? Oh, sorry, my bad. Okay,
Yeah.
sure.
Quiet children.
Yeah.
You go. Shh. Listen. Yeah.
We're gonna slow down.
Yeah. Keep going. Yeah. Round of applause, guys. Round of applause. Okay, all right, let's go to the second part, which you guys, I mean, I feel like it's pretty good.
What do you want?
Okay, so bad.
What do you want?
Main target of AKT.
There's
is
one
bad.
more can
That is BCL2, associated death factor. I don't ever remember what it stands for. I don't think you do need to. When it's not phosphorylated. Oh yeah, okay. So when it's not phosphorylated, it's active. So its job is to hold on to BCL2. Okay, that's its job. When we activate it, it actually lets go of BCL2. Okay, so when BCL2 gets let go of that, it stops programmed cell death. So that's BCL2's job. So its job is to prevent apoptosis. Apoptosis is Programmed cell death, so meaning the cell survives, okay? So, can you go into the next slide?
Yes.
Okay, so when BAT and BCL2 are bound together, the cell surrounded as dies. Why does it die? Because BAT can't release BCL2, and if it can't release BCL2, then BCL2 can't do its job of preventing apoptosis. Okay, so when BAT and BCL2 are not bound together, the cell survives or dies. Survives. Okay, yes, fire photo. So, okay, so only when those two are not together can the cell survive, which kind of makes sense. It's like I think of it as like an alarm call. It's like, all right, it's like the first responder, right? So like they're not together, and he's like, oh man, I gotta go save the cell, gotta go do my job, I gotta be free. That's kind of how I think about it. Okay, questions? Okay.
That we just went through.
Okay.
Okay. Do you want to do this or no? Okay.
Okay.
Is there another one after this too?
Question?
Okay, there's, okay.
Okay, so we did this question last time, I want to do it again, because you're gonna forget about this. So, phosphatases, what do? Do they add phosphates or do they take away phosphates? Take away, that's what phosphatases do, okay? So this guy, P10, takes off the phosphate, that PI3, puts on no, that's, what is that called? That's just PI, right? That's that long name. Would a loss of function in P10 cause cells to undergo programmed cell death and or prevent programmed cell death from occurring, and explain your reasoning? So let's go through this kind of piece by piece. So we know that phosphatases take off phosphates, right? We know that when we phosphorylate PI, it becomes active, which brings PK1, AKT, then that whole thing happens, right? So if we want the signal to keep going, do we want PI3 to have enough PI to have another phosphate, or do we not want to? We want to, right? We want to add a phosphate to keep that signal going. So phosphatases take off phosphates. Why would you want a phosphatase to take off the phosphate that PI3 kinase puts on? You want to turn it off. It's our off switch, right? You don't want that pathway to always be on. So we have a loss of function in P10. So P10 can no longer take that phosphate off. So what do we think that would do? What does that do? What does that mean? No one know it. No one else. Yeah.
Okay,
Next.
So if phosphates would continue to happen, those the cells can die faster.
Exactly. Okay. Yeah. So if P10 can't turn off the pathway, and it turns it off by doing what? Taking off the phosphate, right? If we can't do that anymore, then this would just like lead to an increased active, like a more active pathway, because we're just increasing cell death. Preventing cell death even more. Okay. So if if that phosphate is never taken off, then it's always going to keep bringing AKT and PK1 to there, and they're going to do their thing, and then that signal is going to keep going, right? If we can't turn it off, it's going to just have the pathway stay on. Does that make sense? Questions? Concerns? Anything? Okay. Here we go.
Not that. There's one more. Do you want to do this one, or you just want to move on to JAK-STAT? Do you guys want to do more questions on this pathway, or do we want to move on?
You can do it like rapid fire. Move on.
Move on? Okay. I think it's before this for some reason. There we go.
Oh, okay.
All right, so now we have the JAK-STAT pathway. This one's pretty short, not as annoying as the other ones, but still important. So first, you have your prolactin receptors, which are not tyrosine kinases, they're just normal receptors. So you would have your prolactin, which is a hormone, tied to those receptors, and those receptors will what? Dimerize, right? They'll dimerize. And when they dimerize, they bring the two JAK proteins into contact with each other. And when those proteins are in contact with each other, they cross-phosphorylate. So they stick phosphates on each other. And then the JAK proteins will then phosphorylate the receptor itself, even though the receptor doesn't do anything there, right? So JAKs are tyrosine kinases, but they're not receptor tyrosine kinases. They're just kinases bound to this receptor, which is really confusing and annoying, but that's how it is. So then you have your phosphorylated receptor that the JAKs have then phosphorylated, which leaves a binding spot for stats. And what would stats have to bind to that phosphorylated spot? Yes, perfect. And that's H2 domain. So stats go up and bind the phosphorylated receptor that JAK has phosphorylated. And then JAK can now phosphorylate stats themselves. And once the stats are phosphorylated, they leave the receptor and dimerize. And then once they dimerize, they're able to go on the pathway and get activated by other the active transcription factors for the protein production. Yeah. Any questions on this? Pretty straightforward. Cool.
Oh, we can do this one. This one's a Jack question, the second one. What happened if that's?
Yeah.
Oh, wait, never
oh,
mind.
wait,
No, I lied. Okay. Yeah, I can do this one.
Okay.
Okay, go for it. Okay, real quick revive. What is a gap? Yeah.
One exchange, one is a content exchange factor.
And what do they do? How? What are they exchanging? They're exchanging what for what? Right, okay, yeah, so that's what gaps do, right? So turns it on, exchanges GDP for GTP. Remember, it doesn't always mean when we're going between these two, you guys remember that that D is for like di, T is for tri. It's not always that they're phosphorylating something or that they're taking something away. So in the case of like GPCR, we're actually just letting GDP diffuse away as a whole unit and then just putting in GTP, okay? So make sure that you know that it's not always just phosphorylation that does everything. In this case, it's literally like you're exchanging an entire unit for another unit, okay? I saw that on some quizzes, I saw that on some of the exams. People were saying, oh, well, you phosphorylate this and then turn off, you just take it away. No, in this case, at least we'll get to GPCR in a second, but in that case, it was GDP is just diffusing out because it something changed in the chemicals and it let go of something, and then GDP could diffuse it as a whole. Does that make sense? It's not just like adding something one and then taking it away. Sometimes it's the entire unit diffuses away and something else takes place. Make sense? Okay. What are gaps? What are we doing? If gaps are exchanging GDP for GTP, what are gaps? No. Yeah. Exactly. Yeah. And what does hydrolyzing mean? Yeah, it just like takes away phosphate, right? That's what hydrolysis is. Can we do the? Yeah, okay, so GTPase activating proteins. And these are really interesting. We'll get into in a second. Do I have any more on this slide? Okay, so real quick, let's
You want
do.
this?
Okay, let's just go over this very quickly. Okay, it's a lot easier than you think. It's a confusing diagram. Everyone kind of forgets about it. It's really easy. And I'm going to tell you something that I think a lot of people lost points on this question because they're very small. I think I know what it is. Okay, so when a signal comes in and it binds to the extracellular part of the receptor, what does extracellular mean? Are we outside or inside? So outside, right? When it binds, does G alpha just change like that? No. So here's what happens. Signal comes into the extracellular part, changes the shape, which is conformational change, shape change, and that makes the intracellular part of the receptor change shape, which then causes G alpha to have a conformational change. Okay, so I think a couple of you lost points because some people just said, yeah, signal binds and then GA changes. Doesn't work that way, right? It's a whole sequence of steps there that you're missing. So signal binds, outside of the part changes, which makes the inside of the part change, which then makes GA chain conformation. And when that happens, GA can diffuse away from its brothers and sisters, which are what and what?
Beta and gamma.
Beta and gamma. And it can go down here, it's now active, it can do its job. Okay, so when GA changes conformation, we want it to be active. What do we know about active? What needs to be there for something to be active in this case? GTP, okay? So when GA changes shape, it releases GDP as a whole, okay? So all of GDP is diffusing away from it, we're not just like taking off phosphate. All of it diffuses away, which allows us what to diffuse it? GTP, okay? And when GTP is there, are we on or off? or on, active, ready to go. So it does its job, just does some little signaling stuff. And then it binds the effector protein, you don't need to know effector protein. But GA hydrolyzes GTP and inactivates. So here's what's really interesting, is that GA can actually do this itself. So we'll go over that question in a second, but keep that in mind for a second, okay? And we have these helper proteins called RGS. RGS is kind of what helps us turn it on. We'll get into that. Any questions on like the brief overview? Okay, let's go over this question. I know we did the last one. I know, don't hate me, but like she put it on there a lot, and I think it was it on the exam? Or, like on something?
Yeah.
Guys, you're so welcome. Like that was all. Okay, but I think there's a good chance this may be on the exam. So let's go through it. Somebody remind me, what does a GAP do? You can shout out. Yeah. So what does a GAP do? Hydrolyzes, which means what? What are you exchanging for what? Right. Okay. So basically, both these questions are saying, what are the similarities and differences between RAS, talked about that RAS pathway before, and the GPCR? Okay, and they're different. They're similar in a lot of ways, but they're different in really fundamental ways. So let's go over the similarities. Can somebody give me a similarity between the RAS pathway and the GPCR?
Do you want next? Oh, sorry.
No, you're good. Hold on. Yeah, somebody tell me a similarity between those two. Does anyone remember? I know you know. Yeah, you guys are there. You guys are there at my review session. You remember this one? What, give me a similarity between RAS and GPCR. You remember? What do they both need? Something activity. Guys, I'm disappointed. I'm disappointed. I'll just tell you. I'm just gonna tell you, okay? So both of them need to be turned on, which means they need what activity? GAF activity. GAF activity, right? GAF is we're turning things on. They both need to be turned on. They both need GTP to be turned on. Okay, so you can see the top picture there. It's a circle of like what you need to know. So that's the inactive RAS protein. It gets activated because what is exchanged for what? GDP is exchanged for GTP, right? When GA binds GTP, it's active. When GDP hydrolysis happens and GDP deduces, it's inactive. So it's just basically both saying they both need to be turned on by GTP. That's the similarity. Now the difference. Who can tell me the difference? This was a question been asked over and over again, and wouldn't be surprised on your exam. Yeah.
The alpha can hydrolyze its own GDP, but RAS.
Yes. So RAS has a RAS gap, right? It has a RAS gap. What do gaps do? Exchange what for what? GTP for GDP, which means it turns it off. Okay? So when GDP is there, we're off. Okay? RAS has a guide to help you do that. It has its own RAS gap. GA, to be turned off, it can actually do the hydrolysis, which means taking off phosphate. It can actually do that itself. It doesn't need a gap like RAS does. But sometimes it does. And sometimes we have helper proteins. What are those called? RGS. So in this case, what is the gap for GA? The gap, somebody tell me. The gap. GPCR itself, GPCR, this whole squiggly line, that whole thing, that is actually the gap. Why? Because when the signal comes in, changes the outside of it, which changes the inside of it, which changes the chemistry of GA, which lets it let go of what? GDP. Right? So that makes sense of how that whole system of that signal coming in actually acts as the gap instead of a, like a specific GA gap coming in to turn on. Turn on. Does that make sense? Yeah.
So will these diagrams be on the exam?
No,
No.
these
So-
ones you have to draw yourself.
Yeah, you have to draw it yourself, but you can label everything.
Yeah.
You just can't label like the specifics of what they do, but you can write this down, and I would recommend that you write this down.
Mhm.
So again, recap, what is the gap in GPCR?
GDP.
Yes, but what is it? What's doing that to GA? GPCR itself is the gap. Okay?
This is more.
And then there's a second thing. So yeah, so GA doesn't have a RAS gap. I mean, a GA gap, whatever. It's just GPCR.
Weird.
I said GCPR for that. GPCR. RAS and RAN use a gap to help turn it off.
Yep.
GA doesn't have that. GA can actually do it itself, or if it needs help, it can use RGS proteins to help isolate. These two differences are everything. So number one, know what a gap versus a gap is. Gap means we're turning things on or off. Okay, can do this.
On.
Gap means we're turning things on or off. Off. That's number one. Number two is RAS gets its own fun helpers. GA doesn't. GA just has GPCR, and then if it needs RGS proteins, sometimes they'll use them, but it doesn't need them. Got it? Okay.
Cool.
Yeah, so those are just the key takeaways. And then those like black little lines there, they're just like the lipid anchors, which helps it move laterally. That's all that means. Okay.
One more thing to remember about this pathway is that G alpha is bound to G beta and G gamma, and when GPCR exchanges GDP for GTP, G alpha is able to dissociate from that complex. Yeah. Cool.
Okay, so again, we're not gonna go, we just said this, but I just wanted to highlight that the reason that some of you may have lost points on that question is because you said that the signal comes in and then GA just changes. That's not what happens. What happens? Let me remind you. What happens?
Change in, transmembrane domain, structure, and then in the intracellular portion of the transmembrane domain, and then GTP loses affinity.
Right, which, yeah, so underline star. Signal comes in, extracellular part changes shape, which makes the intracellular part change shape, which makes what change shape? GL. Okay, so don't say signal comes in and then GA just changes. This is her answer key, okay, so this is literally what she said. Don't just say that it comes in and then GA just miraculously like changes. There's a reason that it's going through each step, and part of that is because we really have to highlight that the gap here is GPCR itself. Okay?
Okay, we're gonna take a
5-minute break, go pee, or something, we can get into all that good stuff.
Okay. Cool. Good job.
Like nobody's doing the trunk.
Yeah, I mean, that was kind of expected, but. Oh well. I'm not gonna toss the entire. We should separate them. Okay. Time for skeleton.
This is so funny.
A little walking frame. That's happening right now.
That's literally
In all.
us right now.
That's crazy.
Hope that's on the recording.
What time is it? 6:53. Okay. I'll speed. I might draw the stuff on the board.
You can take your time. You can stay wherever they want.
That's true. I don't think there's anyone else coming here.
Didn't keep track of the time.
Mhm.
It'll just be for
Yeah. I give
Let's
like
be a
another- I'll see, somebody went to the bathroom, so when we come back, I'll just leave them start. Just in front of,
Hello? No, it's fine.
Okay.
I think we can just start.
Yeah. All right, that was not 5 minutes, but we're just gonna start. Okay, now we're gonna talk about cytoskeleton. microtubule, or I guess specifically microtubules, structure and function. Okay, thank you. So we have three main components of the cytoskeleton. I'll just briefly go over the first two. You probably want to be asked about them. We have actin, which is serves as a actin-myosin, which is just another motor protein. And it participates in cellular processes such as muscle contraction, cell mobility, cytokinesis, and organ movement. And it also kind of helps maintain the cell shape and volume. Intermediate filaments kind of do the same thing. They're pretty much just structural. They don't really move. They're just kind of there. And then we have microtubules, the important thing that pretty much everything in this chapter is about. So yeah, microtubules help cells resist compression. They provide a track for vesicles to move through the cell, and that is through motor proteins. Vesicles don't move by themselves; they're transported by motor proteins. And in anaphase, during cell division, they pull replicating chromosomes to opposite ends of the cell, which we got to in topic 8. And yeah, they're also the structural element of cilia and flagella, which is like little things on the end that help them move. Yeah, and they're only in eukaryotes. That's not what I think. Okay. So yeah, microtubules are polymers and tubulin are the monomers. Tubulins are dimers. They're made of alpha tubulin and beta tubulin. Both subunits can bind GTP, but only beta tubulin can hydrolyze GTP to GTP. And beta tubulin has like a basic rate of hydrolysis. It is always exchanging GTP to GDP. Microtubules are polar structures where one end does not look like the other. There's a plus end on one side, and there's a minus end on the other side to the beta tubulin is at the end at the plus side, and the alpha tubulin is at the minus side. Microtubules are also have a property known as dynamic instability, which helps them grow and subtract based on what the cell needs. And then tubulin can only be added to the microtubule in a GTP bound state. You will never have GDP bound tubulin bind to a microtubule. Here's just kind of a basic graph of how microtubules form. You have your tubulin dimers right there. Those start coming together as they diffuse around the cell into oligomers. Those oligomers form protofilaments, and each of those protofilaments start to stack together to form sheets of those protofilaments. As you get 13 of those, they close together into one microtubule, and then more tubulin dimers and oligomers can add to the end of that microtubule to elongate it. Yeah. And then here's just kind of the property of that too. This is dynamic instability, which I talked about earlier. It's the property of microtubules and their ability to be able to constantly grow and shrink. And that happens via tubulin addition or tubulin subtraction. The minus end of a microtubule is typically embedded in a microtubule organizing center, which are basal bodies in prokaryotes and centrosomes in eukaryotes. Let's see. When tubulin addition is greater than GTP hydrolysis, because remember, tubulin has a basic rate of GTP hydrolysis, it's always exchanging GTP for GDP. You will see polymerization, which is growth of the microtubule. So if tubulin is adding faster, then the tubulin below that can exchange GTP for GDP. Your microtubule will begin to grow and you'll form a GTP cap. Opposite, if tubulin addition is occurring slower than the rate that tubulin is hydrolyzing GTP, you'll get depolymerization because there's no GTP cap at the end of the microtubule to stop that. And a GTP cap is just a large section of GTP bound tubulin. Yeah. Okay, so we have our first question. This is talking about dynamic instability. So the microtubule is shrinking because it has lost its what? Yeah, perfect. Because it's lost its GTP cap. If there's no GTP bound tubulin at the end of the microtubule, there's nothing keeping that those tubulin dimers from falling off the microtubule, right? Because tubulin needs to be bound to GTP to stay on the microtubule. How does that microtubule stop shrinking and start growing again? Yeah. Right. So things like that. Yeah. if enough GTP load subunits are added quickly enough, right? Like as you said, if more GTP is adding faster than the GDP is dissociating, then a new GTP cap forms and regrowth can start again. Yeah. And that was on your last quiz, so I would not be surprised if I see that somewhere on the exam. And then all microtubules have, well, not all microtubules, but in the cell, most microtubules have microtubule organizing centers, which are MTOCs. The tubulin in the cell, the concentration is too low to just kind of diffuse around and make microtubules without these centers to stick to. So centrosomes are the thing for most cells, and then basal bodies are cilia and flagella. At the so you have the gamma tubulin ring complex and the gamma tubulin small complex assembly. The minus end of the microtubule is embedded in these ring complexes that sit in the surface of the centrosome. And these basically just help the microtubule have a base to form. And as you'll see later, can reduce the gap phase in those fun graphs that we have to do. So yeah, what's this question?
It's one where they sever in the middle.
Okay,
I can
okay.
answer.
So this one was also on your quiz. This one was,
Want me to do this one?
sure, go for it.
So, okay, kind of a question. I know we don't remember, but from your quiz, there's different answers to this one. So, basically, it's saying that you sever, you're in a lab, and you sever one microtubule in the middle using a laser beam. Would you expect the nearly exposed plus ends to grow or shrink? And explain. Who wants to answer this? A lot of people. Yeah,
They would shrink
they would shrink. Why would they shrink?
because, but the tubulin that's exposed is already now GTP.
Right, okay, so yeah, so answer is it would shrink, right? So again, you have microtubules and then you sever one in the middle, okay? So you're splitting this giant microtubule. So tubulin has the basic rate of like GTP hydrolysis, right? So GTP falling off because it's becoming GDP. And so while the microtubule is growing, tubulin dimers in the middle are hydrolyzing. So we cut it in the middle, okay, fine. The new ends are being hydrolyzed from GTP to GDP. Okay, so severing the tube in the middle would generate new plus and minus ends, right? So neither of which, do they have a GTP cap or no? We just cut it in the middle. Is it just gonna have a cap? No, okay, so you sever this guy in the middle, new ends are created, none of them have GTP caps, it's gonna shrink. So the newly exposed ends are gonna shrink. Actually, I'll just go straight to the answer side. Newly exposed ends would then shrink, and the side attached to the MTOC, which is the plus or minus side. Do you remember? Which side is attached to the MTOC? Minus side, okay? Newly exposed end would shrink, side attached to the MTOC side, which is the minus side, and the other side from the bottom. Basically, just remember, if we cut stuff, new ends are created
That have already hydrolyzed.
that I've already hydrolyzed. And if we're hydrolyzing and we're not adding on stuff, it's gonna shrink. Yeah,
The other side, that's not attached to the MTOC, that side, would that still be growing because it still has the cap?
It would grow from the end at the top that still has the cap, but because it's been split in the middle, right, those, there's still tubulin there that's like already hydrolyzed for GTP, right? Sorry, that's already hydrolyzed GTP and has GDP. So it's gonna start kind of, I don't know,
you wanna draw it?
falling, yeah, sure, like falling apart like a bridge from the middle, right? If you put a bridge in the middle, it's gonna fall down this side and down this side. So even if there's something stable on this side, like the MTOC, and there's something stable on this side, just the GTP cap on the other side, there's nothing preventing this from just kind of falling apart, right? So that will fall apart.
Right, so the ones that we didn't cut, those sides are fine because one of them's already in the MTOC, and then the other one has the GDP cap still. We're talking about the very middle of this thing, which we're just cutting out of nowhere, which doesn't have any protection like the other sides do.
Yes,
Any more questions about this?
yeah,
When it's not cut and they stay together, it's just because they have like a weak but strong enough affinity for each other to stay together, right?
right. So this GTP cap at the end, like GDP bound tubulin still has affinity for each other, just not as much as GTP bound tubulin. And this large thing of GTP at the end is helping stabilize everything down here. But yeah, they still have some affinity for each other, but it's not as good as TP, and it will fall apart if some part of that GTP is lost.
Good?
All right, now we're gonna go to our polymerization assays, which is gonna be pretty much every question about microtubules on this test. And this is when you have free tubulin in the test tube, not in the cell. You would just like have, I don't know, any of you who've worked in lab, seen like micros in the centrifuge tube, you just throw stuff in there and see what happens. So that is all measured by a spectrophotometer, which measures microtubule formation just by shining light through that tube and seeing how much absorption there is. The less absorption, the less microtubules. So in this instance, at time 0, there is no, all the light is getting through, which means there's no microtubules that have formed. There's just tubulin dimers floating around in there. As that begins to form, those microtubules start to absorb more light, and that can be measured with the absorbance on the y-axis. So yeah, so the 3 main phases of these polymerization assays are lag phase, elongation phase, and plateau phase. Lag phase is the time those tubulin dimers take to get together. So tubulin dimers are floating around in there, Brownian motion, all the fun stuff we learned about. It takes time for those to bump into each other, right? They can't just kind of, I don't know, start forming. One might hit another and bind to it, but then, well, it takes more to bind onto that one, it'll fall apart. Who knows? So you take a lot of time, that first in the flat phase, to eventually start forming a microtubule. As your tubulin dimers begin to bump into each other more and you form bigger sections, you have the elongation phase. And that's when microtubule binding is happening at a faster rate than depolymerization. And you have GTP bound tubulin binding to the plus end of your microtubule and growing. And then as that happens, you kind of start to top out, and depolymerization from one end of the microtubule will start to equal your polymerization rates. Or even some microtubules in there might lose their GTP cap and they'll start to depolymerize too. But that's just kind of like equilibrium at the end of the graph where you have polymerization equal to depolymerization. So GTP is binding to some, but GDP bound tubulin is falling off. And that's the plateau phase at the end of the graph. These drugs will all affect different types of graphs. So Taxol is a stabling drug that shifts the conformation of the tubulin subunit so that it has good affinity for the other tubulin in the microtubule, no matter what state it's bound to. So that means basically microtubules won't fall apart. And I think I have a question next. Yeah, I'll ask you about that in a second. Colchicine and vinblastine both do the opposite. They prevent polymerization and cause microtubules to fall apart. Yeah.
We wouldn't need to like have these all memorized, right? She would say what they do.
I feel like every time she's asked questions in the past, she tells you what the drug does, right? Yeah. If it helps, I think the best thing to know is just, you don't have to memorize what each specific drug, how it does stuff, but you could say, you know, Taxol is stabilizing, you know, colchicine and vinblastine are destabilizing, right? So, yeah. So yeah, now let's talk about how we would eliminate a lag phase from our assay. So say we have like our graph and we have, I don't know, this marker doesn't even work.
Wait, try.
Okay. So we have a graph like this, and we have our lag phase. We don't want this lag phase anymore. How do we get a graph that looks like this? Yeah, sure, right there.
You should put an M-top or 2 in there.
Yeah, exactly. So microtubule organizing centers help eliminate the lag phase because they give the place for tubulin to bind. You would need, they give a place while tubulin is kind of diffusing around to bump into, and as it sits there, it stabilizes and more GTP bound tubulin can add on. You get rid of the random motion that happens in the tube, and they have somewhere to organize. So yeah, perfect. Is there another way you would want to eliminate lag phase? Yeah.
I'm not sure if it's like completely eliminated, but yeah. Faster than you're not.
Yeah, perfect, right? As soon as something hits another one, it's just gonna stick together. So you won't see any of those rates of depolymerization. It might not eliminate it like entirely, but it would definitely help. Yeah, perfect. Was there another question too?
Yeah, that was the one, but she answered it.
Okay. Can anyone explain why Taxol would eliminate a plateau phase? Or at least make it start, sorry, it wouldn't eliminate a plateau phase. Why would make plateau phase start higher?
Well, when you don't have the lag phase, that means there's more tubulin available, and so the plateau where, well, one, it stabilizes, so it makes the whole polymerization happen much frequently.
Yeah.
But because there's now more available tubulin, it will be at a higher strength.
Right, well, there's more, I wouldn't say there's more available tubulin, right? Because there's the same amount of tubulin is in the tube. There's more, right, exactly. There's more GTP bound tubulin. So the tubulin levels are the same, but your initial levels of GTP bound tubulin are, there's more of that because there's no depolymerization happening.
Well, if you assume that you don't run out of GTP tubulin, then we'll just have plateau phase, right?
Exactly, yeah. If you had something where you were just constantly pumping GTP tubulin into the system, theoretically, there would never be a plateau phase because it would just keep growing until you run out.
I think a phrase she uses a lot too is like, in these types of questions, she talks about how you can have a non-hydrolyzable GTP analog. I think that's the phrase she uses, and that's definitely an answer. And like that you could use in this type of question, if like you're in a lab and you can just like make stuff, right?
I actually remember that question from my exam. She will probably ask that. Yeah, I would say it would help shorten your lag phase, right? Because if you have more stuff in a tube, it can bump into each other easier. And so it would definitely shorten your lag phase.
But it wouldn't eliminate.
Yeah, it wouldn't eliminate your lag phase. As for your plateau, it could make it a little bit higher. But you still have the same basic rate of hydrolysis happening. So the more tubulin you have in a tube, you're just gonna have a higher absorbance, but it's not because, what am I trying to say? Like if you have the same level of tubulin in a tube with nothing and a tube with Taxol, you'll have a higher plateau phase in one than the other. But if you have one, if neither of them have Taxol, but you add more to one, you'll still have a higher absorbance, but it's not because depolymerization isn't happening, right? Like it's just because you have more tubulin in there. If you have more tubulin in something, you'll have more microtubules, but it's not like because of any different reaction that's happening, if that makes sense.
Yeah, it's just like the rates of things. It's it being slowed down or speed up.
Yeah, the rates are staying the same. So, yeah. Yeah, so.
Oh, I think you actually have a picture of it in the next one.
I think we do. You wanna go?
Yeah, let's.
Okay.
Like that, kind of. In this specific example, I don't think it's the same. But...
Okay, so let's just say we have like 1 molar concentration of tubulin. This one is, let's work this, 2 molar. And this one, let's just say you have a lag phase, elongation phase, plateau phase. Theoretically, in the 2 molar, you would have less of a lag phase, right? You would still have one because there's no MTOC and there's no Taxol in there. But it would be shorter because you have more tubulin on the ground in the same size tube. And that's important to remember, like these would be the same size. And then your elongation would be the same because rates of depolymerization and polymerization are set the same. Oh, perfect.
Thanks, Sarah.
Thank you. That would go up. And yeah, I would say your plateau phase would also be higher, right? Because you have more tubulin in there, so you're gonna end with more microtubules. But your rates of depolymerization and polymerization are gonna be the same, they're gonna stay the same, right? So, yeah. And remember, all of this is absorbance here. So actually, this lag phase would also probably start higher up. The graph just kind of shifts up on the y-axis, right? Because you have more tubulin here than you do here. So technically your plateau phase is higher, but the slopes of everything are probably the same. Does that make sense?
And she will want you to label like the y and x axis of these, and she will grade you on it. So make sure you know like
Axes, these.
what is like what's the units?
Oh, and another interpreter
No, go ahead.
She wants you to draw graphs if you're comparing to a control on the same graph. She didn't take off points in the last one. At least I don't think she did. But typically in science, if you're asked to compare like a control to something with taxol like this, you should draw it on the same graph, not on different graphs, so that they can be comparable. You have like a frame of reference for different things. So if you're drawing microtubule graphs and you're asked to compare stuff, draw them on the same graph, because she might be more strict about that this time.
Yeah, so it literally, like right here, this was exactly how you were graded on exam 2. So she says like 1 point for axes labeling and labeling which line is which, meaning that she wanted them all in one specific graph. So make sure you label those right. So like don't just draw them and then just like expect her to know which one you're talking about. Do every single label, every single axis that you can, because you're just doing them all in the same graph. She's saying 1 point for not having lag phase just because it was referring to that MTalk question about how do you abolish lag phase. And yeah, and then the rest of those two are pretty self-explanatory. So like 1 point for each correct line you have, and then 2 points for the explanation of each of those. So one graph, different explanations. Does that make sense? So what's the x-axis, guys? Time, and then what are you going to label the y-axis? Absorbance.
Yes.
Do we have to do like units for absorbance?
I don't think so.
Okay.
Here's a good example of the graph that you should be looking for on the exam if you were asked about taxol versus the control.
Cool.
Okay, now we have our microtubule associated proteins. This is just gonna be a small, quick section that I'll go through. So you have tau protein and tau dysfunction. These bind and stabilize microtubules and help eliminate depolymerization. They're abundant in the central nervous system. If any of you guys have done research or heard about Alzheimer's, tau is like the main thing thought to cause Alzheimer's when it aggregates. And then kinases can phosphorylate tau and it will dissociate away from microtubules. When tau aggregates and tangles inside the cell, it can cause issues like dementia, Alzheimer's, all that stuff. And now we're getting to our kinesins, kinesins, spun motor protein stuff. These motor proteins walk on microtubules and help transport stuff around the cell. You have kinesin 13, which is a motor domain, where the motor domain is located in the middle of the protein, and it has sliding movement, which is kind of unique. It's like the thing that sits at the end of the microtubule during cell division that kind of rips tubulin off the plus end as you're pulling those kinetochores to the end of the cell. So it's a destabilizing motor protein. Kinesin 3 is involved in cargo transport in neurons. It works as a dimer, which means it has 2 domains, and it is plus end directed, just like kinesin 13. Kinesin 14 walks toward the minus end of microtubules, and it's the only kinesin that walks toward minus ends. So usually you have, I don't know, kinesins, I have no idea, walking towards the plus end, while diamines walk toward the minus end. In this case, kinesin 14 walks toward the minus end.
Yeah. And I would say, like, instead of trying to just memorize every single detail about which one, I would rather have you know the exceptions, because she's usually going to ask you about the like kinesin
Yeah.
14, like that's walking towards the minus end. Like if it's like a short answer one, it's like, which is the only one that doesn't walk towards the plus end? Kinesin, you know what I mean? So like specific characteristics about each of the families, instead of just trying to know like every single little thing,
I'd
because
say
you can't.
the most likely way you're going to see these on your exam is maybe in a free response where you're asked to describe motor protein movement and how that happens, which we'll get into. And if you see it anywhere else, it'll be in a multiple choice. It'll just be like, which one of these kinesins walks towards the minus end? And you know that's kinesin 14. You're never going to have to know, like, this one works in the neuron and like only transports this type of vesicle. She doesn't care about that.
Yeah.
So yeah. You also might have to know like which ones are dimers versus tetramers, but I don't know if she'd be that mean.
I feel like she would.
Yeah.
Or maybe just like a multiple choice question that's like, which one of these isn't a dimer?
Yeah.
Or like, I wouldn't be surprised.
Yeah, that's true. So then you should remember that.
This guy.
5. It's a tetramer. It has 2 tails and 2 heads on each end. So it has 8 total things, domains. It can cause the overlap of 2 different microtubules. You see that in spindle assembly, which we talk about a lot in topic 8. And it stabilizes the 2 microtubules and controls their movement in respect to one another. So it can make them pull apart as they kind of walks to one end of the one on top walks to the other end. Let's see. And then kinesin 1 is just another one that transport cargo. This also walks towards the plus end, as most kinesins do. And it has 2 head domains, and the tail binds the cargo vessel. And can you go back? Oh, and then an example of that is A5, which is, sorry, an example of kinesin 5 is A5, which is involved in spindle assembly. So I would know about A5. That one's probably the only one she'll ask you about, I'd say. Specifically. So now we have the walking mechanism of kinesin. So this one's kind of weird and complicated, but it's really just ATP hydrolysis, which is kind of what everything is. So when you have your kinesin floating around the cell and it contacts a microtubule, that head domain that hits the microtubule already has ADP bound to it. When it hits the microtubule, it changes conformation and ADP dissociates from its binding pocket. And you have an empty binding pocket with a differently, I guess, conformed head domain. ATP associates into that ATPase domain of the motor, which helps bind the neck of that domain to the microtubule. Because you have like the 2 heads, I guess, bound to the things that connect it to the protein as a whole. When that happens, and this first head domain is bound to the microtubule, it causes the back head that's just kind of still floating around because it hasn't bound to anything to be swung forward onto the microtubule, taking its first step, right? And so when it takes that first step, the same process occurs. That head domain that was sitting behind it gets swung onto the microtubule, that contacts it, and that ADP dissociates, ATP binds to it, and then you have that head domain. As that's happening, the one that bound first is hydrolyzing ATP to ADP because it's an ATPase and it just has a basic rate of hydrolysis for ATP. And so that would release from the microtubule and then is swung forward by the first one. Do we have any questions on this? Because that was kind of a quick explanation, but everyone get how this works? Cool, thanks. Oops, is that the answer?
Yeah.
Did I not put a question part to it? My bad.
That's okay, you can just.
So yeah, this is going to be something that she will probably cover on the exam because she loves talking about which proteins are enzymes. She brought it up to us in like TA meetings like 3 times. She's like, should I ask them about which proteins are enzymes? And we're like, no, that's me. And she's like, I don't care, I'm going to do it. So know which proteins are enzymes. Motor proteins are enzymes, right? Because they are exchanging ATP for ADP. And enzymes make and break covalent bonds. Phosphates on ATP and ADP are covalently bound. So they're breaking the covalent bond between the second and third phosphate in the ATP. So motor proteins are enzymes. Take care as to what things are enzymes and what aren't. And just think about making and breaking covalent bonds. Yeah. Do we have any questions on microtubules, motor protein movement, anything like that? Cool.
I can do the next
Sure, I can come up with the time.
Okay, everyone have something to write with right now. Do you have an iPad, paper, something to write with? Okay, super quick. For the next 1 minute, this is from your exam. I want you to rewrite, you're gonna do exactly what you did in the exam. I want just quick memory recall, write down your notebook, iPad, I don't care what you have, type it out, I don't care. Just for the next 1 minute, just a little bit refresh, see what you know, what you don't know. And I'll reveal the answer quickly, but I just want to get this back into your mind as we shift into cell cycle stuff. And remember, she loves checkpoints. She loves checkpoints.
Did anyone answer a question earlier? Like, did you get a peanut butter cup?
Why are you guys being so detailed? I was just trying to get you. Unless you're allergic, actually. That's sensitive.
No one gave you peanut butter.
Didn't mean to offend anybody with a, All right, 20 seconds. This was on exam 2. It's been a while, but we gotta. Cell cycle stuff is like not my favorite unit at all, but she loves this stuff.
She loves everything.
She loves everything. Every single time we're like, oh, I'm not really good about that. She's like,
Yeah.
that's your question.
The only thing she doesn't love is topics 1 and 2, which surprisingly are the easiest.
Okay, I'm stopping it now. Ready? This is the answer. This, I know. I know. Guys, I know. That's why I put up this question. You forgot about this? This was a question on your exam. I wouldn't be surprised if she put it there again, you know, like, well, this was definitely like a multiple choice kind of thing, like, what is the correct order, blah, 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 wouldn't be surprised. And she will give you checkpoint names instead of actual phases because she loves checkpoints, okay? So let's get into it. Phases, what does G stand for? G stands for growth. Cell-specific growth factor signals, check the DNA damage, blah, 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 because what do we need before we're ready to like go into the cycle? Name something, call something else. We need nutrients. We need nutrients. What else do we need? Right, we need a big enough size, right? We can have super small cells. Those are some basic stuff, right? So, mid to late G1. Everybody say that. Mid to late G1. Say it right now. Yes. What do we have? We have the restriction point. Some of you got the timing of the phases wrong on that quiz, which we're going to go over. So mid to late G1, we have the restriction point. What happens when a cell goes through the restriction point? What does that mean? We're fully committed to the cell cycle. I was going to say it. Okay, so yeah, so it's fully committed to the cell cycle because what happens? What's like the mechanism of that? Does anyone remember?
One cycle sequence, then they go and phosphorylate, there's a couple of steps.
Yes, it's RB and what's the other guy? RB and E2F, right? So it's kind of like BAD and BCL2. It's kind of what I think of it like. We'll get into that mechanism, but remember those two terms. So we have S phase, G2 phase. G stands for growth. So G2 phase, we're going to check for completed replication, DNA damage. So there's a checkpoint there. What was the first checkpoint? Restriction point, which happens when? Mid to late G1, okay? That was the first checkpoint. Second checkpoint is what? G2 checkpoint, okay? So those are 2 checkpoints you need to remember. M phase, so that's the prophase, blah, blah, blah. That's kind of boring. But again, another checkpoint in here. So instead of trying to like, yes, know the order of these phases, but even more important is know the order of the checkpoints, right? Because the checkpoints are going to tell you kind of what kind of phase it's in, because they make sense related to each other. In M phase, you have the spindle assembly checkpoint, which check for what? If the what is assembled correctly. Spindles correctly, right? Like it's pretty straightforward for each phase, but she will ask you a lot of checkpoint questions. Or if she asks you a question, she says, like we're gonna go over like the cells get stuck question. You have to say, okay, it's gonna get stuck in this because of what checkpoint, right? So some cells are passing these, some cells aren't. And that's important. Yeah. And then there's the G0 where the cell is supposed to time. So, okay, cell cycle control, CDK. CDK stands for cyclin dependent kinase. What do kinases do? Phosphorylate things, right? Cyclin dependent kinase. That tells you everything you need to know about it right there. Okay, so how do we regulate CDK function? How do we regulate the cyclin dependent kinase? Well, we can adjust the level of cyclin that is going on in each phase, right? Remember how there's like the different cyclins for each phase? There's, you know, G1 cyclin, there's all these different cyclins. We can regulate what that CDK does based off of those, because what is CDK? CDK is cyclin dependent, right? So that's number 1. Number 2 is kinases and phosphatases, right? So phosphatases do what? Take a phosphate off, right? Kinase add stuff. CDK will be phosphorylated by the CDK activating kinase, that's CAC. Okay, so in order for CDK to be activated, we need 2 things. One is we need a certain level of cyclin coming around. and whatever phase we're in. That's number one. What's number two? What else do we need? I just said it. Cat. Okay. So those are two answers. We're gonna go over that question a bit. And then we have CDK inhibitors. So that's, I feel like it's pretty self-explanatory. At microsite near and inhibit ATP or structure state binding. So they're inhibiting CDK. That's pretty straightforward. Okay.
Oops. Sorry.
Okay, yeah, so again, this graph, super important. Different cycling helps CDK target different things. So if I have S-phase cycling going on, it's gonna tell CDK, the main regulator of everything, to do something that the S-phase wants to do, right? So it's just basically like a code, saying, hey, we're in this phase now, we're giving you this specific cycling, because we're in this phase, so I need to do this. That make sense? Okay, so cycling, regulator production, and destruction, so levels of all pretty straightforward. That bottom diagram I would really recommend having on your sheet. So there's an active CDK. CDK is what cycling dependent kinase, and then there's the cycling. Ubiquitination of cycling. Because can anyone remind me what that means? What does that big term mean? Yeah.
Having like ubiquitin group that sends the proteasome.
And the proteasome is what? Yes. So when something is ubiquitinated long term, when I always mess it up, it's being tagged to go to the proteasome. Proteasome is the garbage disposal of the cell. Okay, so you tag something, it's gonna go be destroyed, essentially. So that's how we destruct cycling. Okay? That's just a quick thing. So, quiz question. You observe that CDK inhibitors, let's just start with the first one. Like P21, she loves, I'm telling you this right now, look at me in my eyes. She's gonna ask you about P21 and P53. Guaranteed, okay? There's something she loves. One of them is sigma-7. She loves that. She loves P21, she loves P53. What else is a term that she loves? Everything. mTOR.
Yeah.
She likes them. But I'm telling you, know what P21 and P53 are, okay? So, CDK inhibitors are being expressed and binding to G1 cyclin CDKs. Where in the cell cycle are these cells? Who wants to tell me? Just for the first one.
Yeah.
Yep. Is it during early G1? Is it before or is it after early G1? After early G1. Okay. She's going to be annoying about this. So, okay. So binding to G1 cyclin CDKs.
Wait, can I?
Yeah.
Does anyone know why it has to be after early G1?
Because there's a formation of pre-RCs and they can only happen when there's low levels of CDK, and that's in early G1. So we know then that there's gonna be more of that later on, and that we need that CDK later on, the pre-IC.
Yeah, perfect. So exactly what you said. We need the pre-RC to form, and pre-RC can't form if there's active cyclin CDK.
Yeah, perfect. And that kind of leads into our second question, right? So you observe that pre-RCs are not active forming on origins of replication in the DNA. Where must we be? Early G1. So pre-RCs come first, and then what happens? What comes after? What's the other term? Pre-ICs. Pre-RCs first, then pre-ICs. We need cyclin activity in order to have pre-RCs. We need cyclin activity in G1. Okay? So checkpoints. P53. She loves P53. Okay? So cells check for nutrients in G1. Yeah. DNA damage. Always, right? We have a bunch of different checkpoints for that. Completed replication in G2 after us, and we have proper spindle assembly. Okay? P53 is an essential regulator of stress, and it's a transcription factor. So P53, kind of similar to sigma-70 and like these other transcription factors we've talked about. All it does, this is the only, don't confuse these two. So there's P53, there's P21. P53 can serve as an upregulator for P21. That's its job. Don't think about P53 more than that. Think of it as a way to getting to P21.
It's a
Okay.
transcription factor.
Yes, it's a transcription factor. And P21 is a CDK inhibitor. So this would inhibit the cell cycle from continuing. Okay, so something bad is going on. We need to stop the cell cycle.
Why? Because maybe the cell hasn't grown enough. Maybe there's DNA damage. That's probably the most likely reason. P53, a transcription factor, upregulates the production of P21. And P21 is the CDK inhibitor that inhibits the cell cycle from going on.
Just in general, I think a good thing to think about is also what is a transcription factor, because she might ask a multiple choice question like, which of the following is not a transcription factor? And then list like P53 or sigma 70, but like, how do you do something that is, yeah,
Can you just like briefly explain sigma 70?
I can do that one.
sure.
I get a lot of questions.
Yeah, she loves, guys, she loves sigma 70. Okay, so you guys live in a house or a dorm. I don't know where you live, or apartment. Maybe I do know where you live. I know where Lynn lives. Okay, so you need to do upkeep. So every week, I do my bi-weekly manic clean of the house, and I vacuum and I do my cleaning, and I have to do this every 2 weeks to make sure things are kept up, because if I keep it going for too long, but things get too messy, then I get sad. Okay, so sigma 70, this is the term I want you to remember. It's the housekeeping sigma. Okay, so little things like repairing little damages.
No reason.
Like little damages, little things that a lot of different genes need, sigma 70 does. So like it'll repair little things that a lot of different genes need. Okay, so does it matter if you're in a dorm or an apartment or a house? Does it matter? I have to clean all of them, right? Like all of those require certain upkeep and certain things to do. So same with sigma 70. So across a whole bunch of different cells, it's going to help all of those cells kind of do that upkeep, which means, and this is, she'll probably ask you about this. This is going to be such a term that's so long ago, but you guys remember affinities or how much things like each other? What type of affinity, high, medium, low, medium, high, whatever, do you think sigma 70 has to help a whole bunch of different types of cells in their basic upkeeping needs? Yeah, so like a medium, it will have, I would say medium to high affinity. So basically, it's more of a general transcription factors. Why? Because it needs to help a whole bunch of cells with the same basic tasks, right?
But it specifically binds to the -10 and -35 boxes, which makes it sequence specific, right?
Yeah. So it's sequence specific, but it also needs to be able to help a bunch of different cells that may do different things,
Yeah.
right? Because it's like a housekeeping thing. That's what she'll ask you about. So remind me, what is the affinity of a sigma 70? Yeah. Does that make sense? I would say maybe, like, you can say medium high. In my mind, it's a little bit more medium, just because it needs to be specific enough to be able to bind to things, but it also needs to be not specific, so it can do a whole bunch of different things to different cells. Does that make sense? That's why we're gonna say medium to high. Does sigma 70 make sense? She loves sigma 70. Yeah, the last part, like what color is it?
Like, it has to be specific enough to.
Yeah, so it has to be specific enough to actually bind to things and stay binded. But it also needs to be general enough to go and help different cells with different things. So again, this is housekeeping. So it's doing a whole bunch of little tasks that are different from each other, but every cell kind of needs them, just like how every single place you live needs a little bit of cleaning and a little bit of like upkeep stuff. Make sense? Okay. I'm sure she might ask that offending question. She does look so something.
Are we good on P53, P21, CDK inhibition?
So think of it as like the alarm system. The alarm goes off, P53 is like, we need more P21. And then P21 is like, we need to stop the cell cycle because there's bad stuff going on. Make sense? So like what would happen if there's a loss of function in P21? Is the cell gonna stop? Is the cell cycle gonna stop? It's gonna keep going, and maybe it'll keep going with damage stuff, and that's not good. So if she asked you about like the cancerous phenotypes, like that's important, right? If there's anything that happens to P53 or P21, then we can't do that alarm.
So what does that make P53 or P21? If anyone remembers this. Yes, perfect, an oncogene. It can lead to cancerous phenotypes if something messes up.
Yeah, so like the proto-oncogene and then the chilbo tumor suppressor, right?
Yeah.
Which one of those? So P53 and P21 are both proto. Okay. And then these are all the check-ins. Okay,
Okay, this is just kind of a general overview of our
I think
things.
this is the other one.
Actually, I guess P53 would be more of a tumor suppressor because like you are,
Oh, yeah, you're
you're
preventing.
preventing it
Yeah.
happening.
Yeah, okay, yeah.
Sorry. Because if P53 gets phosphorylated, that's in response to DNA damage, so it's a tumor suppressor,
I think the proto-onco would be the CDK and the cyclins.
right?
Yeah.
That I think.
Yes,
Sorry.
sorry about that.
That's my bad.
No, that was my bad too. I said the same thing. Okay, now we're gonna go over some general G1 events.
Yeah.
So we have, this is just a big diagram, of everything that happens. in
It's
G1.
just a-
It's kind of a mess, so look at that if you want, but I'm gonna explain the next slide
The next slide.
because this one's bad. Okay, so before committing to the cell cycle, cells need to check for enough nutrients in G1, make sure there's no DNA damage, and they need to grow. For this reason, we have a checkpoint, G1 checkpoint, that the cell does not cross until it's ready. And in that, we have, and that's controlled by E2F and RB. So when you have grown enough, when you check for DNA damage, you can then get to this part of the cell cycle, which is the restriction point. RB and E2F control the restriction point. RB is permanently bound to E2F until it's phosphorylated by CDK. And CDK only becomes active when it's ready to reach the restriction point. And so CDK will come along and phosphorylate RB, which causes RB to release from E2F. And E2F acts as a transcription factor to promote the transcription of proteins required for S phase. So in this case, do you guys know which of these things would be a tumor suppressor and which of these would be an oncogene? Okay. Right, yes. So RB is the tumor suppressor, right? Because it's keeping E2F from being active and moving into S phase when the cell doesn't want it to. If we move into S phase before the cell's ready, if we have DNA damage, that could lead to a cancerous phenotype. But because RB is constantly bound to E2F, we're keeping the cell in check. And that would make E2F an oncogene, because if E2F for some reason becomes active, it will start freaking out and moving the cell into phases it doesn't want to move into. And then we have cancer. And we don't like cancer.
We don't like cancer.
Cancer is bad.
I could do the next one.
Okay.
Or you have anything else to say about this?
Yeah, I think we're good on this. Does anyone have any questions about E2F RB stuff?
It's kind of just like bad in BCL2. Like
Yeah.
when it's released, then E2F can go and start DNA replication.
I think the main thing to remember about this, and she will definitely ask about RB, is that RB is always bound to E2F, like before passing the restriction point. The way we pass the restriction point is by phosphorylating RB to remove it. Usually you phosphorylate like something to activate it or cause it to affect something else. In this case, we're phosphorylating RB to make it dissociate from E2F.
Does E2F influence the CDK cycle? Is it a positive feedback loop?
I think it might marginally, because as you are promoting the transcription of proteins for S phase, right, you're also kind of upregulating S cyclins. Yeah, what would you say?
I wouldn't, yeah, I don't 100% know the answer to that, but I don't think she'll. All you need to know for this is just like when RB is phosphorylated, it lets go of E2F.
Yeah.
But I would say for CDK cyclin, unless she tells you that something's happening with those levels, you don't have to make your own guess.
Yeah, so it is actually a positive loop. E2F will activate cyclin E, which will bind to CDK and make a CDK2 cyclin E complex, which will cause more phosphorylation of RB. So it is a positive feedback loop. Okay. Now we have pre-RC formation. What's the important thing to remember about pre-RC formation? Yes, it's inhibited by CDK activity.
So this needs to happen in early G1. That will be on the test, I would assume. Yeah, so the pre-replication complex forms when CDK activity is low in early G1. That's because the cell needs to repress forming the replication complex when it doesn't want to replicate. And if CDK is active, it will phosphorylate all the components of this so that we can't, so that we're not replicating in phases we don't want to replicate then. So yeah, if CDK activity is high, we're phosphorylating different parts of the pre-RC and keeping the pre-RC from forming. That's why the pre-RC forms in early G1 when CDK is not fully active. So in the order of pre-RC formation, we have ORC, which is the origin replication complex, which binds to the origin of replication on DNA. ORC recruits CDC to the origin, and then CDC6 recruits CDT1, which delivers two MCM complexes to origins. MCM complexes are basically helicases. They're not basically helicases, they are helicases. What do helicases do? Yes, they break the hydrogen bonds between DNA and unwind it so that DNA can be replicated. Yeah.
Just so we remember, what are the two things that we need to be true in order for pre-RCs to form? One is about the cyclin, one is about CDK activity. What are those two things? Somebody name the first one for me. Yes, CDK activity needs to be low. What is the other thing that needs to be low? G1 cyclin activity, okay? So those are the two things. If those two things, if we have high cyclin going on, if we have high CDK activity going on, it's not gonna happen. Those things must both be true for things to happen. So just to recap, low G1 cyclin activity and low CDK activity needs to happen in order for pre-RCs to form.
Okay, here's more of the pre-RC. So the ATPase activity of the CD6 allows MCM to be loaded onto the DNA, which are the helicases, unzips the DNA, and then CDT1 and CDC6 leave the origin, so you're just left with ORC and your two helicases. And then once G1 CDK is active, so what phase of the cell, or not what phase of G1 would this be? Yeah, mid to late. So in mid to late G1, you have CDK active and it causes the pre-IC to the pre-initiation complex to form at the pre-RC. This is the loading of DNA polymerases onto the origin of replication, and replication will begin, and this is known as origin firing. When origin firing happens, you are entering the S phase because you're starting to replicate DNA. Not all origins fire at the same time, because you have so many, they don't necessarily all have to coincide at the same time. But after E2F is active, it's phosphorylated everything, it's caused the transcription of everything for S phase, and you have all of these things translated and ready to bind to DNA. You have pre-IC formation, and you have S phase. Yes. Good on that? Cool. Here is our question. When the following mutations cause cells to get stuck in the cell cycle? A, loss of function in P53.
And just to clarify, P53 is tumor suppressor, 21 tumor suppressor, because they stop the cell cycle so that cells can't keep dividing and multiplying, which is cancer.
Yes.
Just to clarify.
And cancer is bad.
Cancer is bad. Boo. Boo cancer. Okay, P53. So let's just start with, would the cells get stuck or would the cells not get stuck? Let's do thumbs up if it would get stuck, thumbs down if it wouldn't get stuck.
Nice. All thumbs down. Yeah, because if you don't have P53 suppressing anything, you are just constantly going through the cell cycle. If P53 can't get active, if there's DNA damage, you're just going to progress through the cell cycle anyways, and we don't want that. Did you have a question?
Oh, yeah, I was just to ask, like, why would it get stuck? Because it wouldn't repair, or it would like still have DNA damage.
Yeah, so it will have DNA damage, but if P53 has a loss of function, there's nothing to detect that DNA damage, right? So
They'll still just keep going.
it'll
even
just
though
keep
it's
going
damaged,
with that DNA damage.
which is bad.
The only reason
P53 stops the cell cycle when it detects DNA damage. But we can have DNA damage and still have the cell cycle going on if P53 isn't working.
And how does P53 respond to damage? What does it do? It upregulates what? P21, and then P21 stops it, right? So they're both responders, and P53 is responsible for upregulating P21, which are both tumor suppressing.
Yes.
Okay, loss function in E2F. Remember, this question is, does the cell get stuck? Does it not get stuck? If it does get stuck, which cycle does it get stuck in? So E2F has a loss of function. What happens? So let me tell you, would the cells, let's do it again. Let's do thumbs up for it does get stuck. Let's do thumb down for if it doesn't get stuck. Okay, we're saying it does get stuck. Why are we saying that? Somebody give me an answer. No. You want an answer? No.
Okay, it would be reducing the proteins that are needed to limit the estrogens.
Right, okay, so remember, it's like bad with BCL2, right? What does ETF bind with? RB, right? And so if nothing happens, they're always bound together. Now, once RB is activated, it lets go of E2F, right? So if we're saying, and E2F is the one that's like, okay, now you're committed to the cell cycle. Now you can go past the restriction point. Now you're all good. I've checked you, you're chilling, right? So if E2F can't go and commit the cell anymore to the cell cycle, then it would get stuck because you just didn't pass the restriction point. How do we know if we pass the restriction point? If E2F is released from RB, then it can keep going. So another answer to this would be, okay, like, or a question would be, what if there's a loss of function in RB? Same thing would happen, right? E2F can't be released from RB, which means it can't commit the cells to the cell cycle, right? That's another question, because they're, together.
Yeah.
And this is G1 phase. And I would specify, when does restriction point happen, guys? Mid to late G1. If you're saying G1, there's so much stuff going on in there, always say where it is, okay? Okay, and loss of function is ORC. Who remembers what ORC is? Anyone? Yeah?
Moves on to the origin notification.
And when does it do that? And for pre-RC formation or pre-IC formation? RC, right? And pre-RCs come before pre-ICs, right? So ORC, you can go to the next one. Yeah, so ORC can't load for the pre-RC form during the early G1 phase, right? Pre-RCs let pre-ICs form, so ORC can't load onto it. Cells will progress at the G1 to S transition because they can't begin DNA replication. Why not? Why not? Somebody just repeat back to me what I just said. I know we're trying. Yeah. Yeah, can't pass the restriction point. That's it. ORC can't load, can't make stuff, can't keep going. But remember, specify when that is. So it says at G1 to S transition. This was from her answer key. She said this. Okay.
That's it.
Yeah, little tips. We already kind of said this at the beginning, but use that 8x11 sheet of paper, guys, and label the proteins. Just has to be handwritten. You can label the proteins. We just can't do like definitions and concepts and stuff, but it's gonna be done. It's gonna be done, guys. Like, it's gonna be done. I know you guys have some of you have Orgo to worry about too. Finish it. Don't think about it again. Whatever you think you did, it's not gonna impact your grade that much in this course, I promise you. Okay, like I really thought I knew my stuff, and then I looked out and I was like so in my bag. I was really sad. But it wasn't as nearly as bad as I thought it was. But make sure, please do those graphs. Guys, like that, those graphs need to be like your religion because she loves those questions. So remember to label. I hope this was helpful. I hope this boosted your confidence a little bit. Email us with any questions. Thanks for staying for 2 hours. We will send this recording out. And I will see, you're also proctoring, right? We will be there at 10:30 tomorrow with you in Tottenham, which is, by the way, a horrible place to take these in, but it's happening. And if any of you are really sad during it and need like a pep talk or something, just call. There's gonna be so many of us, like Please ask questions if there's any confusion with wording, or if you just need like help, like moral support. We're here.
Like the graph. and
Thanks,
like,
guys.
So, like, just for, I want to write down the notes, but like, what would you add to during the lag?
To shorten lag pace,
Yeah.
MTOCs, right? Because you have something that's helping your tubular, like that is giving your plant a tubular to bind. And then,
What was it? So it was that, MTOC.
right, MTOCs will help shorten your lag pace. And then Taxol will as well, because you're not having any dissociation. of genes.
And then, I remember there was something about seeds and then, and then is it only Taxol that's like medicine that does that?
Taxol and stabilizing.
You can be like.
And then Taxol, but the seeds, I remember there's something about like seeds.
Yeah, so if you have like already put together parts of microtubules, that'll also make it faster because they're already.
But it's mainly MTOCs and.
Anything that doesn't let you hydrolyze ATP and stabilizes the microtubule will typically shorten.
Was she asked, like, what would cause the lag phase to be like longer, or does she?
I don't think so.
And then, so question, how would it affect the graph, so like, how would it look compared to a control? When you like,
You'd have a shorter lag phase and you'd have a higher point.
and then why would there be a higher?
Because if you have Taxol, your microtubules never depolymerize, right? And so a plateau happens when your rates of depolymerization are the same. When you never have any depolymerization, that's just gonna go up until you use up all the tubular. Yeah.
Yeah, and then, in terms of signature, I think I get what she's saying about it generally, but like, this is like when we questions were asked. And like, what you think were asked, like, what would you? Yeah, so she was like, because I remember something about sigma stampede and helping to, like, helping the DNA codes like bind onto like onto the DNA and like stay there, have higher affinity with the DNA so that they can, and then the higher the base it has.
Yes, it's already it's the polymerized to the.
And then like, I guess like with that.
Hold on, do you have like the photo, like the diagram of it that she sent out? Hold on, I'm just gonna end this call.
You had it for that relationship, well, like, what should I know besides it?